You've landed at the edge of what medicine has known and what it's becoming. I'm Dr. Richard Rocker, and this is the Integrative Continuum, a podcast bridging biology, energy, and human potential. In each episode, we explore how quantum biology, frequency medicine, and ancient healing systems are transforming our understanding of health. This is where mitochondria meet meaning and blood work meets bioenergetics. Subscribe now and join me on the frontier of integrative healing, where science and soul are no longer separate.
Welcome to the Integrative Continuum. As always, this podcast is about exploring health, medicine, biology, and human potential with curiosity, open-mindedness, and a commitment to standing by the truth as I know it and as I continue to learn it. Today, I'm joined by Mark Lintern, an independent cancer researcher, multi-award-winning author of The Cancer Re-Solution, and creator of the Cell Suppression Theory, or CST. Mark's work has attracted considerable attention because it challenges the dominant genetic mutation model of cancer and proposes a different way of looking at the disease, not simply as a genetic malfunction, but as a suppression-based response involving intracellular fungal pathogens, altered cellular terrain, immune disruption, and failed biological regulation. After his own cancer diagnosis and the loss of a close friend to the disease, Mark spent 9 years immersed in the peer-reviewed literature, developing a model that he believes accounts for all 10 Hanahan and Weinberg hallmarks of cancer, as well as more than 20 additional features of the disease. In 2023, he presented his theory to a panel of 10 international cancer experts before a live audience of over 200 medical professionals, receiving an average confidence score of 7.9 out of 10. His work has since won multiple awards and has attracted serious institutional engagement, including a pilot study in development with the cancer charity Yes to Life and the U.S. biotech company Ultra Botanica. This is an important conversation, not because we are here to give individual cancer treatment advice, but because cancer theory matters. The way we understand a disease determines the questions we ask, the treatments we prioritize, and the possibilities we can even imagine. Mark's work asks a bold question. What if cancer is not only a disease of damaged genes, but also a disease of cellular suppression, altered terrain, infection, immune dysfunction, and disrupted biological communication? Because every treatment approach is shaped by an underlying theory of what cancer is. This conversation has real practical relevance. It may help patients, clinicians, and researchers think more deeply about the assumptions behind cancer care, while always encouraging people to discuss any decisions with their qualified medical and oncology team. It is a pleasure to welcome Mark Lintern to the Integrative Continuum. Welcome, Mark.
Thank you, Richard. It's a pleasure to be here. Thank you.
Great. Yeah, very happy to have you here. As I was telling people in the opening, your book, The Cancer Re-Solution, as you can see in your background and as I have here, fantastic book. I'm absolutely enjoying your book and the way you write and do your research, and highly encourage people to check that out. So, Mark, for listeners who may not know your work, can you tell us what led into your cancer research in the first place?
Yes, it's a very different journey than most. So my background, first and foremost, I'm a graphic designer by trade. And it was a very long time ago, I developed cancer myself. I was lucky I had it surgically removed, but that started me on a journey of research because I wanted to understand which particular DNA mutations had caused my cancer. My GP and my oncologist couldn't tell me. So I just wanted to, try and work out if I could prevent it from coming back. And that was fine. I carried on with life, no problem. But then a friend of mine got cancer, Sam, cervical cancer. And I thought I'd already done a little bit of research. So I'll delve into the research again to see if I can just help her get through this period. And unfortunately, she passed away after a year. But that year led me down quite a bit of a rabbit hole. initially really starting off my curiosity by it opened the door to different theories of cancer and the concept of realizing that actually the origin of the disease is still up for debate. I mean, there are approximately at least 9 credible theories of cancer at the moment. That number varies depending on how much validity you apply to different theories and how you assess them. But there are a number of theories out there, and it is an open-ended question at the moment because none of those theories are proven. And that drove my curiosity because we're told, and I was under the impression initially that cancer was a genetic disease, and it still may be. It's just we're currently at the theory stage of evolution in terms of the scientific analysis of this incredibly difficult and complex problem. However, it's presented in the literature as if we've moved past the theory stage and we've already acknowledged and identified what the origin of cancer is. So we've moved into a territory where we elevate the somatic mutation theory above all other theories as if that's fact. And yet it hasn't been proven, which I think is a serious problem because we're not getting the results that we were hoping for by targeting genetic mutations in the way we have been in the last 5 or so decades. So that for me just emphasized that there was a potential issue there. I mean, that cancer could be caused by something in addition to genetic mutations. They could both work in synergy with something else. But at the very least, it led me down a path of thinking which was that something else in addition must be involved because we're not making as much progress as we would like. And then after Sam's death, I discovered the metabolic theory and various other theories. And I was just very upset at the time. I didn't want to, Sam's death to be in vain. So I thought I could carry on with my research and just document the information I already had just to help anyone else I knew who would possibly develop the disease. They could read this and then realize that there's other things they can potentially do and research to give them hope. But then that led into 8 years of research and a eureka moment for me that made me develop a brand new theory and identify a different mechanism through a different paradigm.
Yeah. It's, you said the standard cancer treatment has gone beyond the theory stage, like we have it all sorted out. And I always think, when I hear this phrase, the science is settled, it really kind of annoys me because I, I don't think the science is ever settled, and we should be asking questions. And through asking questions, we can help to, test those theories out, which you definitely have been tested out with your, what was it, 6 to 8 hours in front of the, the cancer oncologist and the like. So I think that is important that we never forget that. I think you bring that beautifully in your book. You bring a lot of questions out that really makes us think, and this is really what we need. So when we're talking about making us think, you're not originally part of like the conventional oncology establishment or even a medical background. So do you think that helped you to see things differently?
Absolutely, I do. There's a huge creative element to problem solving. That's number one. I mean, Einstein did. I love this quote by Einstein. He said, "Logic will get you from A to B, but creativity will take you everywhere." And there's that aspect of it. And with my design background, it's taught me to really question my thought process. Because whenever we're tasked with a design problem, it is you don't lead with your initial idea that you think and feel would be the actual solution to any particular problem. You force yourself to adopt some more research and adopt a completely different way of thinking because we've got to come up with original ideas. We can't plagiarize any sort of design that's currently out there. So it's a constant mindset of always looking for something new and always challenging your own perspective. And I think that really helped me try to consider something new. I wasn't afraid to kind of make mistakes, because we constantly make mistakes all the time in graphic design. That's one of the reasons why, sometimes we may stumble across something and then that forms part of the design. And at the end of it, we might say, oh yeah, we planned that out, but really we didn't. But it's a creative process. You've got to allow yourself to make mistakes. And I find that in the medical industry, I think a lot of it's built on reputation. And I think there's a fear of making mistakes in the medical industry. But on the other side of that, well, because of that, I haven't got a reputation of any sort in the medical industry to lose. So I'm quite happy to ask questions which may seem rather ridiculous at the time, but these have led me down different rabbit holes that then allowed me to see things from a different perspective. The other thing is, I think there is an issue with a medical education in the sense that it can be institutionalized. And I don't think it encourages necessarily creative thinking as much as it would do in the background that I come from. So you get what I've seen in the research is you get a lot of, obviously medical science is incredibly complex. So I can understand why everything is siloed as the way it is, but these medical disciplines don't necessarily talk to each other. It's almost frowned upon to wander into another area or another discipline. And I think that's an issue because what I've seen is there isn't much crosstalk, and cancer is incredibly complex and multidisciplinary, in my opinion. And there's a huge benefit to be able to not have any fear about going into and questioning the different disciplines and drawing on information on a broad spectrum to see how everything fits together. And I think that's been the advantage for me as well. I'm not constrained to one perspective. I've been able to go where my mind pleases, basically.
Yeah, that's good. There's two things that you said. You mentioned fear, and you always wonder that the people are going to go, oh, what would they be afraid of? And so, do you think is it more of an egotistical thing? Is it a financial thing, or maybe is it combined? If somebody's doing research, we know that often grants are made, and is it if you don't follow along this line, you're not going to get your money? What have you noticed?
Yeah, well, it's a mixed bag. I've come across a lot of really leading scientists who I've got incredible respect for, and they are incredibly humble. They've spoken to me, and they've expressed their gratitude for what I've done, and it's really nice to have that sort of response. But then I've come across people, you can tell the ego kind of dominates in that situation, and they might appear a little bit offended by the fact that I'm from outside the field, and they've got a particular view, and of course there's that reputation side of things. So there might be a little bit of ego going on there as well, but also financial side of things. People have to earn a living, and it's very difficult when you've been, for decades, convinced that your field is correct, and you've been pursuing that avenue, to then question that, you're almost questioning your livelihood in a way.
Yeah.
And that is a scary path to take. So I understand why there's hesitancy. But also it just takes time for people to adapt to new ideas as well. So there's that. And especially I think because I'm from outside the field, there is that extra amount of hesitancy.
Yeah. It's it's almost like that. I can understand being in your shoes, because you will get some people where the ego comes into it, and they go, well, what do you know? Who are you to tell me this with your background? And what I found going through my medical career, if you want to call it that, is the people that I've learned the most from, and I got the most beneficial information from, has always been the researchers. It hasn't been the doctors. It's been the people that are in the field that are digging in and researching. They're the ones that are figuring it all out. The researchers, the scientists, and they're just telling the doctors what they found. So it depends who you're listening to. I think that plays a big part into it. Yeah. So, and if we look at modern day uh, doctors, a lot of them are so busy, they don't really have time to research. So they might be just looking at headlines, or uh, they might just be looking at the drug rep who comes by and gives them maybe a very one-sided look at something and not outside of that. And I think that happens a lot in these days, and I'm sure you've come across that multiple times. Yeah.
Yeah.
Um,
I think it's the system. It's just it needs to change because it doesn't work in the favor of the doctors because, like you say, they're far too busy to be able to spend the time to do the research. So that does need to change.
Yeah. And the other thing that you mentioned about things being siloed, this is something I've referred to many times, that essentially medicine has become very reductionist, and we have all these hundreds and thousands of specialists that are focusing on a minute aspect of health or a minute aspect of a cellular function or that, and it's great, it's fascinating, I love it, but who's taking all of that stuff, who's putting it all together to make a system that works, right? And and this is what I really like when I've been reading your book there, and I see you building up that perfectly, taking not just one theory but multiple theories, and finding, hey, where is the answer here? What can we do that actually is going to help people here? So that's great. Um,
Yeah,
so I suppose,
just on that point. Sorry to interrupt.
yeah, go ahead.
Um,
Yeah,
that was the issue that I came across when I discovered the different theories of cancer. It's really important to be looking at different things from different perspectives. But what I found was the proponents of different theories were quite staunchly in favor of just pushing that particular theory, and it
mm,
seemed to get to the point where it's not about presenting truth. It's almost about sometimes just trying to prove that that theory is correct.
yeah,
And what I've tried to do is, I was on board with the metabolic theory, and I am still on board with the metabolic theory. It's the most it's the most established theory with the most explanatory power I can see at the moment. However, there were still some contentions with that theory, so I couldn't wholeheartedly jump in and champion that theory alone, and that's what led me down this different path. So I've always tried to, and because I've not got a medical background, I've had to rely on the evidence. So I've had to
mm,
be as objective as I can be, and I've had to take a stance of, asking questions and never stating anything as fact, because that's not how science works anyway. That's how I thought
yeah,
science operated. But so I've tried to follow that principle, and I'm not saying with my theory that I'm particularly right. I'm just saying, and this is what I thought scientists, and most scientists are like this. But I'm just trying to say that all the evidence is out there. I've done a broad look at the evidence, and a huge bulk of this evidence is pointing down an avenue where is that's been overlooked. We haven't really looked at this particular section. It just seems as if everything's pointing towards this, and it can almost beautifully explain the disease almost in its entirety.
hmm,
Now, again, that's not saying that I'm right. It's saying that here's many correlations that almost turn into a bit of a signal, really, when you put them together, that's worth investigating. It's absolutely worth investigating, and that's where I leave it,
hmm,
because that's, to me, that's what science is. You need to bring other people on board so they can investigate it further and find the holes, if there are any holes in it, and then there's this to and fro, isn't there, um, between trying to find out what the actual truth is. I think
yeah,
that's really been what I've been trying to go for. It's not, I don't really care if I'm right or wrong. In fact, um, if I'm wrong and I try and push my particular view, that's not going to benefit me. It's not going to benefit my family and friends or anyone around me,
yeah,
because I'm going to be pushing something that could be damaging to them.
yeah, absolutely. Well, as with anything in life, we learn the most from our errors and mistakes. I don't think everybody gets it right all the time. It's this is what it is. That's how it works. So yeah, absolutely. Okay, let's get a little bit into some of the theories. So for decades, cancer has largely been explained through the somatic mutation theory. So the idea that cancer is primarily driven by accumulated DNA mutations. So what do you think that model explains well, and where do you believe it may fall short?
Um, very good, very good question. It's what does it explain well? No one's actually asked me that question before. It explains well, I would say. Um, there are a number of common mutations that occur, and those we see that there is an effect with those mutations. We see a huge increase in risk through those mutations. And there are a number of one or two drugs that have provided a quite substantial benefit. Gleevec, for instance, for non-Hodgkin's lymphoma. But by and large, I'm more in the field of it doesn't really explain anywhere near as much as I would like it to.
Mm.
Because the impression I'm getting is that it is the theory that explains what cancer is. However, the more I've researched, the more it doesn't seem to explain. So there's a huge issue I find with that particular theory. Again, it could be that cancer is a genetic disease, but it's just far too complex for us and our technology to fathom at the moment. So that might be something down the line.
Mhm.
However, the evidence building against the theory, the contentions that exist are numerous, and they shouldn't be ignored. So that needs to be looked at. So one or two things just to highlight, and that is that there's been tissue found with cancerous mutations, the healthy tissue with cancerous mutations, and those mutations don't give rise to cancer. And then you have tumors that do not harbor any of the expected driver mutations at all. So the question there is what in particular is driving that particular type of cancer if it's not going to be those mutations. There's also a recent study of around 7,000 tumor samples that identified around 1.7 or 2 mutations on average, which is far too few for cancer to develop. And then you have the nuclear transfer experiments where they transferred the cancerous nucleus into a healthy tissue, healthy cell, and we didn't get an abnormal growth. But when you transfer the cytoplasm into healthy tissue, cancerous cytoplasm, we then get cancer growth. So that suggests that it's not the nucleus, it's not the DNA within the nucleus that's causing this mechanism of abnormal growth. It's something in the cytoplasm. And then Professor Paul Davies mentioned this a while back in 2013 in his presentation of the atavistic theory. He highlighted that around 80% of the studies that support the somatic mutation theory cannot be replicated. That is, they're incorrect. So a lot of the science being produced and what we think is correct about the disease is later retracted because it cannot be replicated. So there are huge, huge contentions. And one more final one would be the randomness of DNA damage that occurs. So we have a consistency to the disease, which is the hallmarks that occur. So each solid tumor follows the same pattern. And this was a big thing for me. The metabolic theory actually identified this consistency with the Warburg effect that occurs in all cancers or thereabouts. But with genetic mutations, we're expected to believe that utter randomness, not just from patient to patient, but from within the same tumor. The heterogeneity is quite incredible within the same tumor. So the question is, how could so much random damage to the DNA cause the consistency of the disease even within the same tumor? It doesn't quite add up from a consistency and a problem-solving perspective. So all those combined, it led me to really start questioning the established view.
Yeah. And why do you think this mutation model has remained dominant for so long?
Consensus, I think. Well, I think initially the idea was good. It it was very plausible. It started in the 1960s, just after Watson and Crick identified the structure of DNA, and it was very plausible. It's very plausible to think that the mutations, because there are plenty of mutations in cancers, those mutations could drive the disease, being as though DNA pretty much can drive all different mechanisms within the cell. It's almost the blueprint, you could say, for want of a better phrase. So naturally, a lot of funding came in through that route, and it's the God particle, isn't it? It's the ability, it's the excitement of possibly amending how or creating designer DNA to eradicate disease. I think initially would be, but there's other things that you could do with that that are exciting. So it just garnered a lot of interest, a lot of investment. And when you have a huge amount of investment, you essentially have a juggernaut. Then consensus follows. And when you have that juggernaut of investment, it then becomes hard to actually question that paradigm. So it becomes the established paradigm, especially when your career depends on it. Your earnings depend on that, and it must be very hard. I would really respect the scientists who haven't gone down the genetic route and are looking at the metabolic theory, the cancer stem cell theory, and the different theories, tissue organization, field theory, because there's less money in that field. So it's a combination of those things, really.
Yeah, yeah. Okay, so we're talking about mutations. So how do you distinguish between mutations being a cause of cancer versus mutations being a consequence of cellular stress or disease progression?
That's a great question. Well, as far as I'm aware, there's different types of mutations, and in particular, scientists have identified the driver mutations, which they categorize as the ones that are actually driving the particular mechanism of cell proliferation, of apoptosis. So there's specific detailed hallmarks that drive angiogenesis, for instance, that actually enable the tumor to grow. There are many other side mutations, of course, that may affect the ability of the immune system to function. But generally, it's the driver mutations that are the key mutations that we're looking, or we should see a pattern in, but we don't. We see that they're random
Hmm.
and non-existent in quite a number of cases.
Yeah, yeah. So, okay. So, do you think with modern oncology that it's become a little bit too much focused on the tumor itself rather than the biological terrain that allowed the tumor to emerge? I know this is a big contention between various groups.
Yes, I think it's got to the point where we can't see the woods for the trees.
Hmm.
So terrain for me is absolutely everything.
Mhm.
That's health. That's mitochondrial function. That's cell signaling. That's just your body working as it should do to repair, regenerate, and working at its optimum. As soon as you start compromising that in any particular way, not only are you causing damage that can then be copied into other cells and generate the wrong type of proteins, the loss of signaling, but you're also opening the door to additional problems such as infection into the wound, and it just complicates things. So yes, it's terrain for me is everything. And what I felt was when I started initially doing my research was that scientists, yes, they're siloed, but I felt that they'd done enough research across all uh, aspects of the medical spectrum that we should be able to identify if we step back and look at it from a distance what the patterns were. So that's really where I went down that particular avenue, and I think that's what really needed to be done. No one's really stepping back and looking at it holistically across the board to find connections. And yes, I do think the problem is that because things are so complex, we can focus on the really minute aspects of the cell itself, even biophysics. That's another realm in itself. But if you do focus without coming back, then you lose sight of the overall picture, and I think the overall picture really matters.
Yeah, absolutely. So you've developed or coined a phrase, the cell suppression theory. So for somebody who hasn't heard about it before, can you explain that in simple terms?
Yeah, I'll explain it through cell malfunction, just the
Okay.
difference between. Uh, so basically, I I believe there's a fundamental flaw in the way that we're looking at cancer. When you look at all the other theories, established theories, say there's 8, they fundamentally make an assumption that the cells malfunctioned, and then every reasoned question and avenue of investigation follows from that assumption, because every theory is looking at what part of the cell is damaged, what part of the cell is broken, and how that is generating the disease. So
Mm,
the cell is malfunction. That's the problem. It's gone rogue. It's now no longer part of the body. So we've got to attack the cell. I believe that's an issue, because when you focus on the cell and blame the cell for being the problem, that prevents you from looking elsewhere at other external effects. And one of those effects I'm suggesting is pathogens. Now, so I've put forward a paradigm which is actually that our cells aren't going rogue and they aren't malfunctioning. They're actually doing what they should be doing, and that is responding to a stubborn intracellular infection. This switch that we see in the Warburg effect in the energy system is a response by the cells, by the infected cells, and the cells around the infected cells that aren't actually infected. They're all collectively working together, and they're responding, triggering the Warburg effect in order to target the pathogen. And the issue is the pathogen is sustained within that tissue for
mm,
various reasons, because the terrain is damaged far too much. It's allowed the pathogen to gain a foothold, and in that moment, the immune system has been suppressed because of the damage to the tissue, the iron overload that occurs, and the lactic acid that occurs. So we have this element where the pathogen locks in the Warburg effect because it's sustained within the tissue, and the immune system and the infected cell itself and the surrounding cells aren't able to actually eliminate the pathogen. And because the pathogen is sustained, we have this lock in this process, this proliferative state. So for me, it's the pathogen is suppressing the pathways within the cell and the tissue, surrounding tissue, in order to survive. So it's blocking the cell death mechanism. It's forcing the cell to shift to this Warburg metabolism, because it's suppressing certain defensive mechanisms that the cell is trying to establish in order for it to survive. So that's where the suppression aspect comes from. Because I want to make a little distinction, because when I do talk to other clinicians, they often say, well, infection, we accommodate that anyway, because viruses and bacteria, we highlight that they cause cancer. But when you look at those particular theories, they're not talking about the novel mechanism of suppression, by active suppression by the pathogen that's hijacked the cell. They're not talking about that. They're
yeah,
talking about the pathogen damaging the DNA. So the virus damages the DNA, the bacteria damages the DNA, and then the mechanism or the origin of cancer is the DNA mutation or is the mitochondrial damage. That's the mechanism. I'm saying that's not the mechanism. The cell is trying to do what it should do. It's not the enemy. We should be supporting our mitochondria to target the actual pathogen itself, not attack the cell. And if I may go on a little bit further, just to illustrate my point with the immune system is having a really difficult time trying to identify the tumor.
mm,
And immunotherapies have a really difficult time too. I mean, some work really well, but not for everybody. And then you get hyperactive disease in others. So it's a double-edged sword. And the question is, why is the immune system not able to see the tumor? And there's a couple of other mechanisms, PD-L1 upregulation and various other things. However, one of the main points I'm making is that a tumor is not completely not every cell in the tumor is infected with a particular pathogen. You only need a small number of infected cells to elicit a huge response in the surrounding tissue because they all work together. So non-infected cells will switch to the Warburg effect. And then what you have is these cells are locked into that Warburg-like metabolism because it is an anti-infection response. So you have a load of thousands of regular cells doing what they should be doing. They're not the enemy. So the immune system doesn't see them as the enemy because they're actually trying to eliminate the pathogen. They're regular cells doing their regular job. So that's one of the main arguments I put forward for highlighting why the immune system has difficulty in identifying and attacking the tumor, because in effect, you're asking it to attack self. So yeah.
Yeah, yeah, it's interesting. There's so many thought processes and theories in regards to tumors, and I've heard many things that it's a sac, and it's a sac that may contain toxins, or I've heard that it may contain some kind of infections in that, which means that you would want to be very careful about damaging that sac, because if there was an infection in there, then all hell could literally break loose, right? And I suppose that's why when they're doing surgery on certain tumors, they go, well, this is just simply too much of a risk here.
Mhm.
So then with your theory, then if there was an infectious process, then wouldn't it make more sense to treat the infection to make the environment less habitual for these infections?
Yes. So that's primarily the approach that I would argue that needs to be taken and added additionally to whatever metabolic approach, or if chemotherapy, whatever it is you are using, off-label drugs even. Because one of the main things I've found in the research, only really because I'm looking at it from this perspective, is that you look at the number of off-label drugs that are being used and trialed and tested, and they're all also antifungal. So the additional property they have is that they're all antifungal. So I'm finding this coincidence across all different aspects of infectious behavior and the treatments that we use. That's worth looking into. So I'm not saying, there are different drugs will work in obviously different ways, and they can be directly targeting the metabolism of the cell. However, there's this feature of all of them being antifungal. And so the question I would ask is, are we inadvertently having benefit because we are actually targeting the fungal pathogen that I say is potentially driving the disease? Now, that's not to say it's going to be as easy as that and use, say, itraconazole, a broad spectrum antifungal drug, and you're going to clear up the cancer. There's a number of issues because the immune system is already primed not to identify fungal pathogens, for one. And you need the immune systems with a drug like itraconazole because it's a fungostatic drug. It doesn't kill fungi. It inhibits the fungal pathogen. It's not fungicidal. So what you need is the immune system to help out. So they both work in unison. But what we find in pretty much all cancers is there's a switch from a Th1 response to a Th2 response, which is the Th2 response is primarily for cell repair and targeting external pathogens such as viruses outside the cell. The Th1 response is immune cells geared to targeting intracellular pathogens. So we have this mechanism at play where you have the wrong type of immune cells surrounding the tumor, and they're trying to just repair the tissue that's constantly inflamed, and they're not targeting the pathogen inside. So we need a way of, if we're going to target the pathogen, yes, that's what absolutely we need to do. We need to make the environment hostile to the pathogen, reduce inflammation, possibly reduce its fuel source, primarily glucose, but it can adapt to fat and protein as well, which is why there's this metabolic flexibility we'll talk about. But we do need to target the pathogen, but it needs to be in a nuanced way because it's not as simple as just giving them antifungal drug. There's resistance to those drugs. Not all fungal pathogens are targeted with those antifungals as well. So we need to modulate the immune system. There's a number of things that need to be done all in one go.
Yeah, yeah. Yeah, it's interesting when you talk about the fungal aspect of things. the first time I came across research was by an Italian researcher, I'm not sure if I'm pronouncing it right, Simonocci, or he
I
wrote a
don't
book
know.
called
I think it's Giulio Simoncini. That's how I've
Yes,
always.
yeah, that's probably it. Yeah, my apologies to the Italian people out there. But yeah, he wrote a book called, I think it was called Cancer as a Fungus, and that was an interesting read there. So if we look at that theory, um, and I know from when I'm working with patients that if somebody would have, let's say, a fungal infection, a big part of it is changing the environment. And the first thing that always comes up is, well, what drives fungal growth the most in the body? And it tends to be sugar, right? So we always go, well, you need to go on a low carb or a no carb diet. And how have you found that in your research and work?
Yes, there's definitely a correlation between reducing the amount of sugar intake you have, normally synthetic, refined sugar is the problem, and the same with infection. So if you look at infection studies, whenever you do a fast of any kind, you reduce the fungal burden within the body. This is a
Mm,
well-known phenomenon, primarily because they feed on those sugars. And the problem as well is the biofilms that they generate. So it's very difficult to target them when they're within those biofilms. So that's why I would argue that obesity is linked to higher rates of cancer. Apart from the fact that you have glycation occurring in environments where you have a lot of glucose within the bloodstream, which is going to cause its own problems. But mainly because you are feeding these pathogens over a long period of time, they're establishing themselves within tissue. And then if you've got constant inflammation, those pathogens are sitting and waiting for that opportune moment for the signals of that inflammation in order for them to transition into a pathological state, the hyphal form, and invade cells and tissue. So they're incredibly intelligent, and it is about it is largely about sugar. But having said that, there is research that highlights that just you can't just starve the body of sugar, because your cells need sugar, your brain needs sugar, and the bloodstream regulates the amount of sugar you have within the bloodstream to a specific level. I think the issue is not the fact that your blood is still supplying sugar to the body. It's the fact that people are over consuming the sugar, and then that allows the tumor, because its metabolism is such that it's relying on glycolysis. It actually requires, in order to generate more cells, more glucose to produce more of the metabolites needed to generate extra tissue. So, and simply because glycolysis absorbs a higher rate of glucose for every ATP molecule it produces. So yes, cancers definitely require a higher rate of glucose. We see this with the PET scan, and that feeds into the survival of pathogens within the body. So the two go hand in hand, in my opinion.
Hmm. So then, so then with that logic, if we look at different dietary types, then there's an awful amount of positive research on a ketogenic diet, and being in a state of ketosis can help a lot of people with very serious conditions. So how would that play into a situation like this?
Yeah, you see, the research does seem to show that there is a very good benefit, and even with chemotherapy, because you're strengthening the immune system as well. Because one of the other things I found was, because the surface structure of fungi can be primarily carbohydrates, 90% carbohydrate in structure. If you consume a lot of glucose and carbohydrate, it's flooding through the body. The neutrophils actually find it difficult to differentiate between the carbohydrate structure of both. So it can suppress the immune system in that sense. But sorry, can you remind me of the question again? I went off a little bit of a
Yeah,
tangent.
we're just talking about a ketogenic diet and being in ketosis and how that may help the environment, the terrain, and being able to help somebody with, let's say, a metabolic process, whether it's obesity, cardiovascular disease, cancers, as the big top three.
Yeah, so if you want to look at it really simply, and you'll know better than I do, but there's essentially two states within the body, an anabolic and a catabolic state. And the ketogenic diet gets us into that catabolic state. So the anabolic state is when food is abundant, like in a business. The economy is booming. You take on a load more employees, but then you get to those employees. That business grows to a size where you actually have a number of jobs that aren't really doing much. And it's the same with cells in your body. You're accumulating, you're storing fat, you're storing more food, and you're generating more cells because the times are good. But then those cells become maybe senescent, or they don't function as well, less mitochondria, or the mitochondria don't function very well. So when you enter into a state of ketosis, you enter a catabolic state, or you're trying to get yourself in a catabolic state. And that's like in the business world when the economy dips and there's less money going around. So you have to, what's the word? You have to economize. You have to make your business more efficient. So you get rid of the lesser efficient stuff, and you reduce things down. And it's the same with the body. So we have autophagy, mitophagy occurring. And this is where the body actually recognizes that there's less food coming in. So in this state of fasting, you could say, or a ketogenic state, the body is trying to conserve energy. So it breaks down the inefficient cells and uses those nutrients to improve the mechanisms, the functionality of the rest of your tissue, your immune system. And then you have mitochondrial biogenesis occurring. So you have this process of renewal effectively in your mitochondria, in your immune system, and you eliminate a lot of the inflammation that occurs in the body because you're getting rid of those cells that generate the inflammation because they're not working as efficiently as they could be. So that in itself is a fantastic, you could argue, medicinal process that allows your body to regenerate. But also it's cutting back the glucose that is available for the tumor, and it's cutting back the glucose that is available for pathogens to survive and thrive within biofilm and within the tumor itself.
Yeah. So then really the modern day food pyramid, which is slowly being changed, is really not serving us in that process of helping to build the terrain at all, really. When you look at I know the classic U.S. food pyramid, I think they had 6 to 11 servings of grain-based carbohydrates as a base of a healthy diet, which somebody who studied nutritional biochemistry, I just think that's funny, or ridiculous funny, but I don't see how anybody can build a proper terrain with a diet like that. And even when it comes to digestibility of foods, we have to be able to digest and break it down to assimilate any benefits from that. So I think the whole diet wars out there right now has got a lot of people confused. And as I say, a lot of people are looking at philosophical ideologies instead of looking at biochemical sense on how things really are.
Yeah,
So,
well, I expect, you look back at our ancestors and they will have been in ketogenic states at certain points and fasting at certain points because they couldn't actually get access to the abundant food that we have now. So the body goes through this cycle of this catabolic cycle it needs to go through. And that makes it more metabolically flexible and efficient. So, it's a natural process. And I think we've lost that because we've got access to food 24/7 whenever we want.
yeah, absolutely. And yeah, and to so many different foods, if we can even call some of them foods. I've heard people saying, "Oh, you need to have such an array of different foods." And I remember one person saying it was an unreal number. It was something like, "Oh, you have to expose yourself to like 200 different foods." And I'm like, I don't even have 200 foods at my doorstep unless you want to include processed and ultra processed, which I don't really would call foods to begin with. So I think we've made things a little bit too complicated. And I think diet and nutrition is actually a lot simpler than people believe it to be. And
Yeah, so the
yeah.
ketogenic diet isn't just about, starving the cancer of glucose. That's the point. There's other mechanisms going on within the body that are regenerative and beneficial on a number of different levels. So it's not just about the sugar. But the other thing is the ketogenic diet can be very good and very beneficial, but it's not the answer to everything. And that's what kept me moving forward with looking beyond the metabolic theory with a number of contentions that were there. But like, for instance, prostate cancer, you would want to be careful about how you diet with prostate cancer because it isn't driven by the Warburg effect initially. So this is one of the big contentions with the metabolic theory. It's actually driven by oxphos and fatty acid oxidation, or it appears to be. Glycolysis is the base metabolic rate of healthy prostate cells is actually an upregulated form of glycolysis in conjunction with fatty acid oxidation. So yes, there's a different explanation. I think I'm the only person who's actually provided an explanation for prostate cancer because I show that the abnormal metabolism of prostate cancer, which doesn't map onto the metabolic theory Warburg paradigm, is easily explained through infection. So that's the beauty of what I'm saying there. But just the point is, you can't just go and say obviously ketogenic diet is beneficial for all cancers, because it's not. You've got to understand the nuance and the base metabolism of any particular cancer at any given moment in time. Most of them are affected in that way. However, there's just one or two. Breast cancer, I think, is another one.
Yeah. So based on that, different cancers may react differently. So does your cell suppression theory apply to all cancers, or do you see it as more relevant to certain types than others?
No, all cancers. So
All
Rowan
cancers.
Straussman has done the research in 2017, and he overturned the notion that tumors were sterile, free of microorganisms. So he found that bacteria and fungi exist intracellularly within all cancers and all tumors. So that's the first big thing. The second big thing is that the cell doesn't appear to be broken, malfunctioning in the sense that the metabolic theory suggests, in that it's reliant on glycolysis. So it's just reliant on glucose and glutamine, effectively. That's the argument there, because a lot of people are showing, a lot of scientists are showing, and there's a lot in the literature showing that you can actually upregulate oxphos, and certain cancer stem cells are operating using oxphos. And like I say, so is prostate cancer. So it seems as though the cancer cell is metabolically flexible. So that's one big question mark over the metabolic theory. But also, that's got to really factor in if you're thinking about what treatments you're going to be utilizing. And the way I explain that is that the pathogen within the tumor can actually adapt. It can adapt to glutamine and fat. It upregulates particular pathways, and it does this when it gets absorbed by an immune cell. It rapidly upregulates, say, the glyoxylate pathway, which allows it to convert fat into glucose. The pathogen's always trying to convert back into glucose. I mean, there are a number of pathogens that are lipophilic, so they'll consume fat, such as malassezia, which has been implemented in pancreatic cancer and breast cancer, and a number of others. But primarily, they're glucose loving. But if you do cut off the glucose supply and the pathogen isn't eliminated and the immune system isn't able to get in there, what you then have is a cell working on autopilot, because that's what cells are doing when the pathogen's in control. The pathogen has blocked the cell death mechanism. Now the cell cannot die. So it's literally just trying to rebalance, because our tissues are constantly always trying to reach homeostasis. So the pathogen is absorbing the nucleotides, the glutamine, whatever compounds are inside the cell and in the surrounding tissue in order to thrive itself. And the cell is then forced just to balance those off. So if the cutting of the glucose isn't quick enough, or the pathogen can easily adapt, and the cell is also absorbing glutamine through substrate level phosphorylation, to allow ATP to occur, then the pathogen will sequester that glutamine and convert that through gluconeogenesis into glucose. And the same with fat. So it's understanding that there is this metabolic flexibility, and the pathogen being present and in control of things explains why that is able to occur.
Hmm. Yeah, yeah, yeah. It's fascinating when you look at it, and I think you explained that very well with the glutamine, because I think there's some misconceptions there as well. When people go, "Oh, that's an amino acid, so therefore maybe we shouldn't be eating any proteins." And is there any truth to that, or have you found that the diet type varies depending on the stage of cancer that you're at?
Again, I wish there was an easy answer. It's incredibly nuanced because, I mean, you can check the metabolic profile of particular tumors at the stage that they're at, can't you? But there's sufficient evidence to show that actually, if you supplement with glutamine, that can have a beneficial effect depending on the stage of the cancer
Hmm.
and what its metabolic profile. So the reason for that being, the glutamine is supporting the stomach lining, repairing the gut. So you're reducing the inflammation going into the bloodstream from the gut and the toxins that are going into the bloodstream from the gut, because you're going to have a dysbiotic microbiome.
Yeah.
And it also, the immune system relies on glutamine as well, heavily, in order to function properly. So by supplementing glutamine in certain circumstances, and I'm not a clinician, so please do not take any of my instructions as medical advice, you can actually benefit the tumor. Benefit the patient and reduce the tumor, because arguably the tumor will only absorb a certain amount of glutamine. It's not going to increase exponentially the glutamine if there's more glutamine available. It can only grow at a certain speed. And then the pathogen can only access the glutamine to a certain degree. But if you're also enabling the immune system, and if you're able to then transition from a Th2 to Th1 response, then, and you're making the immune system, you're optimizing it, because the problem is a lot of patients are glutamine deficient because when the tumor is actually absorbing a lot of glutamine, our bodies it becomes semi-essential, because
Hmm.
the amount that we're producing is not enough to sustain the optimal function of the immune system at that point. And that's when you start to have a problem, really. So yes, the nuance is you can actually optimize the immune system with those amino acid, or that amino acid in particular. So it's not a one size fits all. It's really trying to understand the nuance.
Yeah, yeah, absolutely. And we very much, especially in functional integrated medicine, we always talk about healing the gut. And like you said, when there is a dysbiotic um, stage that leads to kind of more of a full-fledged intestinal permeability, that you can't really get somebody better until you heal that. So there again, we have to look at, well, what causes the intestinal permeability? What causes the dysbiosis in the first place? What lessens the mucosal lining to protect from leaking into the bloodstream? And yes, there can be pathogens there, but then there's also the inflammatory reactions, right? It's again, we're back to the foods that we eat, the toxins that we're exposed to, the medications that you're taking, the stress that you're under. And who doesn't have stress in this day and
Mm.
age? They're all loads of stresses. So it's always a multi-layered approach, isn't it?
Oh, absolutely, absolutely. The body's an ecosystem, and at the heart of it, I think, is the microbiome. We don't touch on it
Mm.
enough. I mean, the microbiome produces a number of metabolites that we actually require. It removes the toxins that we absorb, can
Mm.
do effectively. The bacteria also combat the pathogens, if so we keep the bad bacteria and fungi in check. But this is the other thing. Part of my research was showing that if you have a dysbiotic microbiome, not only are you getting inflamed gut and leaky gut, but candida within the gut, when they overgrow to a certain potential, they can cause inflammation and disease to occur in a different part of the body. So there's one particular study which was showing that they caused a mass amount of inflammation in the lung and disease in the lung. This was directly from interacting from the gut. So you've got to realize that they the health of the microbiome, or the health of the human being, really follows the health of the microbiome, in my opinion. Because I mean, there's been studies showing that fecal transplants, for instance, appear to be quite effective in a number of diseases, and helping the microbiome come back to good health. But just simple studies that are done in rats. When you look at you feed a particular one set of rats a particular diet on the same diet to another particular group of rats, and they have a different microbiome, you get obese rats being generated, and other rats, they're perfectly healthy in terms of weight. And then you switch the microbiomes over, and they switch their weight dimensions. And that's purely down to the type of bacteria that are present, that are feeding off of maybe the glucose that is present. So less of it is being absorbed by the person themselves. So we still don't know enough about the microbiome, but what we are learning is pretty incredible, and it's integral to our health, as well as looking after our hormonal balance and everything else within the body. It's an ecosystem.
Yeah, absolutely. Yeah. I think we're just scratching the surface on what we know about the microbiome and bacteria and probiotics and the like. And I think it's almost like this big messaging system that affects the signaling inside of the body, essentially. And when we go back to the diet, um, what throws the gut out the most? That's what when I work with patients, I'm going, well, how's your digestion? And are you bloated? Are you gassy? And if so, what have you been eating? What do you notice when you eat that triggers that? And I think listening to our body is something that we don't do enough. Or sometimes some people are ignoring the message that it gives to us, and we have to kind of tune back into that again. Yeah. Um, You mentioned the microbial environment. Let me just get this in my head. So there's a microbial environment of the tumor. So could that be one of the missing pieces in why tumors behave differently between patients? May act one way in one person and different in another.
Yes, I believe so. So it's not going to be one fungal pathogen, and it's a combination of viruses, bacteria, and parasites working
Yeah.
together, because you never have an infection of one particular microorganism. That's it. They work in synergy. So that super complicates things. Plus, different fungal pathogens um, will operate differently, and they'll produce different metabolites. They'll affect DNA differently, and it's that complexity of the multitude of DNA mutations that occur, the damage to mitochondria that occurs. So, I mean, there is obvious damage happening to the cell. But if you're damaging a random array of chromosomes or DNA genes, you're going to get really a plethora of random responses, random proteins being produced that are just going to go on and cause further inflammation in all different organs and different areas of the body. So yes, for me, it's the pathogen that's causing a lot of the randomness, because you've got to imagine that this has been going on for a long time. The pathogen's been present for a long time, and it produces reactive oxygen species. The cell's producing reactive oxygen species. It's producing genotoxic metabolites. We have acetaldehyde, all sorts of mycotoxins are being produced in order to eliminate the immune response and damage particular aspects of DNA. So that's been going on for a very long time. And that's why it's random for me. And that's why things can operate or seem to appear different from one patient to another, even though they've got the same cancer, and even within the same tumor of the same in the same person as well.
Mhm. Yeah. Um, so going back to the mitochondria, so do you see mitochondria dysfunction as a cause, a consequence, or both?
I think it's both. However, I'm moving more towards 60%, 40% with the microbial interaction. So
Hmm.
mitochondria in the terrain need to be damaged to a degree that enables the pathogen to take control. Whether or not mitochondrial damage on its own could actually be the driver, I'm open to that. However, there's a number of studies that kind of challenge that perspective. You could argue senescent cells are the result of mitochondria becoming dysfunctional to the degree that they don't really work anymore. But oncocytomas are benign tumors, and they literally have this completely dysfunctional mitochondria, so they can't utilize, and they can't utilize oxphos. But
Yeah.
because they're reliant on glycolysis, as the metabolic theory suggests is the underlying mechanism that drives cancer, the mitochondria are dysfunctional, so reliance on glycolysis drives proliferation. That doesn't occur with oncocytomas, which have really dysfunctional mitochondria. So there is a question there for me: how dysfunctional can the dysfunction of mitochondria actually drive this mechanism? Because mitochondria are also involved in apoptosis, the cell death mechanism. For me, it's working out at what point would the dysfunction to mitochondria prevent apoptosis from happening? Because as far as I'm concerned, past a certain threshold of damage, apoptosis is triggered. And that would combat the fact that the mitochondria are dysfunctional past a certain point. Because I'm even arguing that senescent cells and those benign tumors, they have a small microbial constituent that is affecting those tissues. I think in terms of the oncocytomas, that the pathogen has damaged mitochondria so much that it's actually now detrimental to the functioning of the tissue, so that it doesn't grow. Because there's a number of papers that highlight that oxphos is required, a certain level of oxphos is required for proliferation to occur. So I'm gearing towards, really, it's more, and I'm putting my flag in the ground here, it's the pathogen that's actually hijacking the cell and forcing the shift to occur, where mitochondria are focusing on trying to combat the pathogen. So they're not using the oxygen to generate energy, because they are allowing glycolysis to do that, to generate the ATP. That allows the defensive mechanism, the mitochondria, to then target the pathogen, utilizing that oxygen indirectly and directly. So in a long-winded way, I'm edging much more to the fungal pathogen being in control. And when you kill the fungal pathogen, that relinquishes control, and then the mitochondria, regardless of how damaged they are, will either regenerate from healthy mitochondria being fed in from healthy tissues in the periphery, or they will trigger apoptosis because they've been damaged past a certain threshold. And we see this. This is one of the other things, one of the other contentions, is that we see that if you give natural compounds to cancer cells, they trigger apoptosis. So honey or even silver, it triggers apoptosis, which suggests that cell death mechanism isn't damaged beyond repair, which is really what the metabolic theory is suggesting. That's why it shifts over to glycolysis, because and the cell doesn't die because mitochondria are damaged too much for that to happen. But quite clearly, it can be instigated, which suggests to me that there's a suppression or a suppressive mechanism going on, and that would be, for me, the pathogen blocking that cell death pathway.
Okay. So then,
Excuse me.
to kind of sum this part of the topic up, so from a systems biology perspective, what for our listeners, what are the upstream conditions that would make cancer more likely?
Anything that generates chronic inflammation, damaging the tissue, weakening the immune system. Because I was always trying to explain the consistency of cancer. So you've got a number of different carcinogens. They all damage cells and tissue and DNA in completely different ways. Really random, yet they somehow manage to generate the consistency of the disease. So what is the consistency with all these different carcinogens? And for me, it was four things. Weakening of the immune system over a prolonged period of time, chronic inflammation, which opens the pores to the cell and the tissue, which allows fungi to gain greater access. Lactic acid overproduction, because whenever you've got inflammation, the cell switches to the repair mode, which requires glycolysis. So you've got overproduction of lactic acid and iron overload. Past a certain point of inflammation, the cell and the tissue cannot store enough iron for any length of time, and it gets released. Now, the lactic acid, and the iron feeds the pathogen. It's like both are like rocket fuel. The pathogen senses the local environment. When it senses enough lactic acid past a certain threshold, it realizes that's almost telling it that we have an inflamed situation and the tissue is vulnerable. But at the same time, the lactic acid and the iron overload suppresses the immune response at the site of damage. So you've got this niche where pathogens can take advantage of this inflammation. So it's anything that generates, doesn't matter what it is, which is why there's so many different things appear to be able to cause cancer. And that's not the same as an origin.
Yeah.
The origin is the fungal pathogen, I'm saying. So the fungal pathogen gets into the cell. You provide it with that opportunity to access the cell because of the prolonged inflammation that you are providing it with. Whatever toxin, pesticides, lack of nutrients is generating the weakness to the immune system and the tissue. So the tissue is no longer able to function optimally to resist the invasion. But the key thing is the pathogen needs to be present in the tissue as well in sufficient number. If the microbiome's okay and there isn't that pathogen present, doesn't matter if you're going to have the mutations or you have the carcinogens, you're not going to get cancer. And that is how I also explain the inconsistency of that, because you can still take a number of these carcinogens or you can have the particular DNA damage, but it's not 100% guaranteed that you're going to get cancer. For me, it's because you haven't got the fungal pathogen present in sufficient number within the tissue at the time, or you have. That it's the pathogen that differentiates the inconsistencies in what we see between patients.
Yeah. You mentioned iron overload. We hear a lot about iron these days, and it seems to be a very debatable topic for a lot of people. So, is there truly an iron overload, or would you say there could be more of an iron transportation issue?
A bit of both, I think, because iron is highly reactive. When there's a difference between transportation and iron being freely available. So I think it's the freely available iron that I'm talking about.
Okay.
So, excuse me, with inflammation, you have a process called anemia of inflammation. So the body knows that iron is highly reactive, and it also knows that iron is like rocket fuel to pathogens. So it instinctively sequesters iron away into iron proteins, storage proteins, and the immune system does that too. So the whole goal of that is when you have an acute infection, you remove the iron, which could facilitate infection, to then allow the repair process to establish, eliminate the pathogen if the pathogen's present, and then you release the iron because it's required for cells to grow. Mitochondria, many different aspects of the cell require iron. So in that acute process, that's fine. But after a long period of time of prolonged inflammation, you cannot sequester the proteins that absorb the iron to sequester it away so the pathogen can't get access to it, become saturated. And the tissue can no longer hold it. So now you have this freely available iron, which is damaging, and the pathogen can get access to. And the pathogen requires the iron in order to grow. So, like I say, it's like rocket fuel. So in that situation, iron's bad in the sense in chronically inflamed tissue. So it's a dual-edged sword. You really need iron to survive.
Yeah.
But too much iron in a situation where you are overloaded with inflammation and you have a high pathogen fungal burden, that's going to have a negative impact. It's going to become pathological.
Yeah. So do you think for somebody in that situation, do you think that, let's say, going down to the local blood draw place would be a benefit, giving a pint or two of blood?
That's interesting. That's a very good question, because what we see is there's a difference between men and women in terms of how often they get cancer and who's more likely to die from cancer. It's men.
Mhm.
And one of the many reasons of that is because women obviously lose a certain amount of blood each month. So yes, there's this factor that the iron is less available and it's being used more efficiently in the female body. There's other issues as well. The X and Y chromosome, I think it's the X. I can't remember which chromosome it is now, but one of them, I think it's the X, codes for immunity. So men have a reduced or less aggressive immune system because of that, where women have a more aggressive immune system. And that's one of the reasons why women, I think they suffer a little bit more from autoimmune disease. But that more aggressive immune system can also eliminate the pathogens that I say are driving the disease, which would translate into why women have a reduced rate of cancer and a reduced rate of dying from the disease.
Yeah. Yeah. So I guess the guys, we got to fight a little bit more and get a few more bloody noses, don't we? Little
That's one
fight
way of
club.
doing it.
uh, um, what's your opinion? And I have an idea what you're going to say, but your view on conventional cancer treatments such as surgery, chemotherapy, radiotherapy, immunotherapy, targeted drugs.
Surgery is one of the best interventions we currently have, isn't it? It accounts for the bulk of the survival figures that we have. So yes, I would, excuse me, always argue for surgery where it's needed. However, again, there's always a nuance there where there's the danger of releasing whatever contents of the tumor into the bloodstream.
Yeah.
So you would need an adjunctive, treatment response for that.
Yeah.
Radiotherapy, again, I'm not an expert on this, but I think this damages the tissue more than it should do. It raises, I think, glucose levels. It does a lot of damage that can be ongoing and can, suppress the immune system. So I think that tips the balance in favor of the pathogen for me. I think it can work in targeted certain situations where it needs to be, but I think it's more damaging potentially than it could be. And similarly with chemotherapy, chemotherapy has its place. And Dr. Robert Gatenby highlights that he's been looking at reducing the dosage of chemotherapy and showing that low dose chemotherapy can actually work really, really well for longer periods of time with less toxicity.
Yeah.
And I mean, we can always combine these things with chemotherapy. But for me, chemotherapy, while it's very good at targeting the regular cells, and it's good at shrinking the tumor initially. So it might be really good for shrinking the tumor initially, and then it can be resected, removed via surgery. That can be a good strategy. However, for me, the prolonged use of chemotherapy, I think, is an issue because you're damaging the mitochondria, which are your defense mechanism. You're reducing the quality of the immune system. You're damaging tissue further, causing further inflammation throughout the entire body. And it targets fast dividing cells. Cancer stem cells are not fast dividing, and they have efficient efflux pumps and the ability to pump out toxins. So what often we see occurs is the tumor comes back because it develops resistance. Why does it develop resistance? It shrinks dramatically, so it looks great, but you have this element of cancer stem cells that then regenerate the tumor because they have to regenerate the tissue a lot quicker than before because they're associated with that dying tissue. But also, part of the way that chemotherapy works, in my opinion, and it's documented within the literature, is that it produces a lot of reactive oxygen species. And it does that by damaging mitochondria. So it's the same with any toxin entering the cell, isn't it? Mitochondria will react and produce reactive oxygen species. Excuse me, I might have to get a drink of water in a moment.
No problem.
During that process, as you're damaging mitochondria in every successive then chemotherapy treatment, you've got fewer mitochondria that are efficiently able to produce enough free radicals. So my argument is that initially the free radicals that are generated en masse by the initial chemotherapy is actually able to combat and kill the pathogen, the fungal pathogen present, or at least weaken it. But then the fungal pathogen upregulates its antioxidant mechanisms. The cell is doing that as well because the ROS is so widespread and damaging. The mitochondria are being reduced. So the next phase of chemotherapy and the next progressive phase of chemotherapy, fewer mitochondria producing fewer reactive oxygen species, while the fungal pathogen is upregulating its antioxidant defense. So it gets to a threshold where you can't produce enough free radicals in order to combat the pathogen. And then, of course, you're stimulating the cancer stem cells.
Yeah. Yeah. Um, yeah. So, historically, many important shifts in science become really, as we said in the beginning of the podcast, is uncomfortable questions. Do you see your work in that tradition?
Asking uncomfortable questions.
Yes.
Absolutely.
The biggest shifts in science began with uncomfortable questions.
Absolutely. Because the uncomfortable question really is, have we misunderstood the underlying cause of cancer? And are we, like a juggernaut, are we pursuing a path that is not as effective as it could be, if I want to be politically correct? So the uncomfortable question is that the majority of the cancer industry are on this juggernaut. And at some point, they've got to ask themselves the uncomfortable question is, am I actually, on the wrong path here? Do I need to transition to a different, probably more difficult path and be really honest with what I'm doing? Is it not going to get us to the goal that we want it to get it to? And do I need to get off this train? But that's a very, very difficult thing for a lot of people on that train to ask themselves. So yes, it is a very difficult question, and they are the questions that do change things, because unfortunately throughout history the consensus has been proven to be wrong. It's the only way that progress is ever made.
Yeah. So then what would need to happen for your theory to be fairly tested?
A lot more funding, for one, in this area. There are a number of different studies that can come from my research. At the moment, I do have a biotech company on board, and we're looking at doing a pilot study, an observational study, and hopefully that will grow into something else. But there's a lot of testing that can be done with different pathogens and how they relate to cancer and how it functions, and then the progression of hopefully different antifungal drugs potentially. Not just antifungal drugs or other off-label drugs, the combination use of those, and natural compounds. But we all know that that's very difficult to actually get the studies on natural compounds, because there's not a lot of money in that can be made from those. But at the very least, antifungal drugs, because we're going through an epidemic, really, or even a silent pandemic of antifungal drug resistance. And that's something that needs to be addressed, because it's becoming a real big problem. So a lot of these antifungal drugs we currently have aren't really working because of that. So to generate an antifungal drug that is both anti-cancerous as well, I mean, that's a very interesting prospect for any pharmaceutical company, I would have thought.
Yeah. And I think maybe aspects that we haven't touched on is going more into kind of the quantum medicine realm, where we're dealing with light and frequencies and the like. And we know, especially with things like red light therapy and that it has a lot of benefits to mitochondrial function. So I would assume that that's a whole other area that could be explored a lot more than it currently is.
Absolutely. I think it's paramount. I've been listening to a lot of Jack Cruse's work.
I love Jack.
Yeah. So I agree entirely with a lot of what he's saying, and the mechanism, how near-infrared light works, being as though it penetrates deep within the body. It's very interesting. What I draw a correlation with is that fungi actually control and manipulate nitric oxide mechanisms, and that's a mechanism that mitochondria use to control the flow of oxphos. And what we find is that fungi can manipulate that pathway, and potentially they're using that to block the complex 4, I think, of the electron transport chain. But where near infrared light comes in is it's able to knock off the bonds, the bonds that the nitric oxide have within complex 5 that allows mitochondria to function more efficiently. So for me, it's about making mitochondria more efficient, which is why you have methylene blue and hyperbaric oxygen therapy and any other particular therapy that improves the functionality or tries to improve the functionality of mitochondria, but especially if it's antifungal as well, because I think that's really going to benefit the process. Because really what we're trying to do here, and the paradigm shift I'm putting forward, is we want to be supporting the mitochondria to eliminate the pathogen, rather than trying to force the mitochondria to do something that we think they don't want to do, or damage the mitochondria to stop them from working entirely. It's a supportive role.
Yeah. In my clinic, we use we have a red light therapy room. We use that quite a lot. I use it Monday to Friday because I'm indoors all day and I got to get my dose of light. And if the weather's nice on the weekend, I'm out all the time. Also, infrared saunas. We use that a lot with patients here. We get really good results with that. And going a little bit more out of resonant frequency device as well that uses proton emission, because that helps to get inside of the cell. So again, that's all dealing with frequency. So this is all the quantum medicine world that we're getting into now, which I think is interesting. I think it's old technology that was maybe used in a different way back when, and we just kind of modernize a way to do it. But yeah.
Yeah, it's very interesting. It's an area I'm also will be adding to my second edition. But blue light, melanin within the body, and it's just very interesting how we see, and Jack points this out, how we see fungi growing in and around Chernobyl, and how they actually use melanin as a virulence factor. So one could argue, when we're talking about how mitochondria produce light, and potentially light being a mechanism of communication within the body and a mechanism of facilitating mitochondrial energy production, one could argue that on that premise, the fungal pathogen is essentially stealing, if you will, the light energy that our cells actually use through regulating the melanin that's absorbing that light wavelength.
Yeah. It's very interesting, and geez, we could go on for a long time about that. But yeah, I look forward to your. Did you say your second edition or your second book?
Second edition of the book.
Second edition, upgraded. Okay, excellent.
But that's not going to be for a little while yet.
Yeah. So, okay. So one of the most powerful things about your work is that it gives people a different way to think about cancer. So how do you balance hope with responsibility?
Great question. It's never about trying to say that my work can affect a positive outcome. There's no guarantee without anything at the moment. As I said at the beginning, we're at a stage where we're still at the theory stage.
Mm-hmm.
But for me, it gives hope because it offers additional conversations patients can have with their oncologist or their medical team that's looking at other particular options. And that's just, it adds to the toolkit, and that can only ever be a beneficial thing, because what we're seeing in the literature is that antifungal drugs are now being used and studied for their anti-cancer properties. So that might be because they're targeting the metabolism of the cell, you could argue, but there's always that antifungal aspect that I'm highlighting. And what we see, what view between my theory and the metabolic theory is that both the metabolic drugs are antifungal and the antifungal drugs are metabolic. So we're both really pushing down the same path. And the nice thing about my work is that I'm saying that we're at the cancer theory level of understanding, in scientific research at the moment. There's no one theory that is correct, but there is a difference between the accuracy of different theories, and you can measure the accuracy. So if you're a patient coming into this and you want to, okay, there's 5 theories or 6 theories that warrant you looking at them. And the reason why you should look at these theories is because each theory, associated with each theory, is a different treatment. Treatments are developed from cancer theory.
Yeah.
So if you're ignoring a particular theory, you're ignoring the treatments associated with it. If you're ignoring the metabolic theory, you're ignoring metabolic treatments. So if you're ignoring my theory, you're ignoring the treatments that are focusing metabolically, but also antifungal as well, that seem to have some efficacy in certain situations. So I'm arguing, how do you differentiate that? All that information. Well, you can use the hallmarks of cancer, the 10 hallmarks. There's now 14, and I think a couple of months ago, Hannah has said some, at least 9 more. So it's getting quite convoluted, I think. But there's 10 at the moment that are officially accepted. Excuse me. And what every theory is trying to do is trying to explain them all. The more you can explain, the more accurate the theory is likely to be. So you want to look at, and I've documented this within my book and my work, and I assess the theories and how accurate they are. So you have, unfortunately, the somatic mutation theory can explain around just 2 of the 10 hallmarks, which indicates everything that we've said. It's a difficulty in explaining those hallmarks. There's no pattern of DNA genes that can explain every single hallmark. It's random, and so that suggests that low of a hallmark count suggests that genetic mutations may be involved, but they're probably not the main driving mechanism. So you don't want to be targeting. You don't want to be using just those treatments. And then you have the tissue organization field theory, which is looking at all the information within the tissue. You want to reduce your inflammation as much as you can because that's a precursor to cancer. It's a precursor to the fungal pathogens invading the cell. That can explain around 5 of those hallmarks. So that, again, suggests that there's a mechanism that is intrinsic to the disease. So we should be targeting inflammation. We all know that. And all the oncologists are aware of that anyway. So that's really not a given.
Yeah.
But then you have the cancer stem cell theory. That explains about 5 too. Then you have the metabolic theory. That explains about 7. Professor Seafield would argue all 10, but I've got contention with 3. So that explains about 7. And then my theory, actually the first one to coherently or better explain all 10. Plus the additional 4 that have been added, I can explain those too, along with a load of other features as well. So from that perspective, as a patient coming in, you don't want to disregard the somatic mutation theory because genetic mutations will have an influence over how your tumor acts. But you want to consider the other theories, and really you want to consider the higher the accuracy in terms of explaining the hallmarks, those theories up front, and consider in combination treatments that are associated with them, targeting those particular mechanisms, because those theories suggest that they're targeting something really specific and significant to the tumor. So why wouldn't you target those? So use cancer theory as a practical tool to guide treatment decisions. I would ask oncologists to do that, and obviously patients always to converse with an oncologist or a medical professional.
Yeah, absolutely. So if you could change one assumption in cancer research tomorrow, what would it be?
That cancer is a genetic disease, period.
There we go. Yeah. Okay. And then, so let's say in the next decade of cancer research, what do you think needs to be focused on?
I think we need to equally, as much as we can, focus on the other theories. Those 5 at least I've just mentioned, mine, cell suppression theory, metabolic theory, tissue organization field theory, cancer stem cell theory, and we've still got to look at the genetics, so somatic mutation theory as well. It's just share the resources around, because there's so much evidence in support of these other theories that we're potentially missing something big that can really help and make a big difference in the near future if we were to provide those resources.
Yeah. So for people who want to know and understand your work properly, what would be a good starting point for them?
Okay, so I have a website called cellsuppression.com. That's where you can get most of my information from. I also have a Substack publication called The Cancer Vigilante. So I bring up articles on there. My most recent article is about prostate cancer, explaining the abnormal metabolism of prostate cancer, which is something that hasn't really been done before. So those two are a really good starting point, and there's many different podcasts I've linked to on my website. I have a free PDF, which is a synopsis of my theory. It's a very short explanation in layman's terms of what my theory is all about. So by all means, go on there, download that, understand the basic principles, provide that to your oncologist, see what they think, and then the book's available there too.
Excellent. Excellent. Yeah. And we're going to put all that information into the show notes there as well. So, while you were talking, I was scribbling a few things down here, and I want to kind of give a summary. I'll see what you think of my summary here. All right. So the body is not random. Cancer behavior like chronic pain, fatigue, autoimmunity, or metabolic dysfunction, it may be a logical biological response to altered inputs and altered cellular conditions. Theory determines the treatment, and if the theory is incomplete, treatment strategy may be incomplete. The terrain matters. So tumors do not arise in isolation from immunity, metabolism, inflammation, oxygenation, microbial ecology. What else do we do? Hormones, nervous system signaling, environmental exposures.
Stress.
And overall, patients need clarity and not fear.
Yeah, brilliant.
Sum it up?
Absolutely.
Okay.
Yeah,
Good
nicely
stuff.
done.
Good stuff. Well, Mark, listen, it's been a real pleasure to have you here. I think we skim by so many things. There could probably be a 2, 3, 4, 5, 6 parts. But again, we really highly encourage the listeners to check out Mark's book. It is really great. I love reading, and your book is really good. It really captures my thought and imagination and the possibilities that can arise from that. So definitely encourage people to check that out. And as always, please like, share, subscribe, especially to those that could benefit from this information. And that will really help to get the word out there. So again, we'll put all Mark's info in the show notes. And we wish all the listeners a good day. And until the next time and the next podcast, have a great day. Take care. Thanks, Mark.
Thank you. Take care, everybody. Thanks, Richard.
Thank you for joining me on the Integrative Continuum. I hope today's conversation gave you new insights and practical tools for your own health journey. If you enjoyed this episode, please subscribe, share it with someone who would benefit, and leave us a review. It helps us spread this message further. Until next time, I'm Dr. Richard Rocker. Stay curious, stay empowered, and keep moving forward on your path to integrative health.